Well, thank you for the invitation, Ronsi, and the opportunity to be here and tell you a little bit about some of our work in the lab. I have to say, actually, this work was inspired by Amir Jazari, who's in the audience, a gynecological oncologist. I believe he spoke yesterday. And when I first came to Houston about 5 years ago, I actually had a family friend who developed ovarian cancer. And I was interested to learn more about the disease and understand, what us as bioengineers could do to help think about new innovations and new solutions. And I also have to give a big shout out to Amanda Nash back there taking some videos. She's the PhD student at Rice who really did all the work. So I feel like they just did it mostly, and I just kind of kept telling them that, hey, we got to work fast, because if this works, we want to get it into people as fast as possible. So, just real quick, some disclosures on the work that I'm going to present today is the basis of a spin-out company, Avenge Bio, which is based up in Boston and is the company that has this technology approved for clinical trials beginning this fall. And so just a little introduction about what my lab does and what we think about. So when we look at what the pharmaceutical companies are developing, they're developing a lot of biologic type drugs. And biologics are difficult and complicated to administer. What's fascinating is that biologic drugs are made by cells just in giant bioreactors and factories. So my vision when I started my lab was, can we think of innovative ways to bring that manufacturing right into the patient? And we thought about ways where we could create these little microfactories that housed cells. Let's see if this pointer works. Yeah, there we go. So these little bioreactors that are 1.5 millimeters sphere particles. Inside, there's engineered cells, and they can be programmed to produce a wide array of biologics. They can sense their environment and adjust dosing. And so our vision was to think about ways where we could deploy this technology to make it easier on patient to really deliver drugs where it's not possible, and to ideally develop more effective therapeutics. And everyone in this room is very familiar with the difficult statistics associated with ovarian cancer, and particularly for advanced cases, stage 3 and beyond. When there's recurrence, there's not a lot of options for patients, unfortunately. And when I talked to Amir about, what things have worked, he pointed us to this trial, which was done actually in the early 2000s by Edwards, who's a gynecological oncologist at the University of Pittsburgh. And he actually did several different trials. And what he was thinking, his hypothesis was that there are these drugs, in this case interleukin-2, which are molecular messengers that activate immune cells. Now, these are really difficult to deliver. When you inject them systemically in your blood, you have a lot of immune cells. And so you give this really potent immunoactivator, all of a sudden you activate all your immune cells in the blood, create a lot of crosstalk. The immune cells aren't exactly where they need to be to attack the cancer. So we thought about, and I think Edwards was inspired by this same sort of mismatch, and he considered, what if we just infuse in the IL-2 in the intraperitoneal space where it needs to be? And so he hooked up these patients to like a pump system and infused in the drug. The drug is actually unstable and difficult to formulate, so he had to administer a couple of liters a day of fluid. Many of the patients couldn't actually, take the therapy, but in the several patients that actually, out of 24 patients, there was 6 responses, 4 complete responses, and 2 partial ones. The limitations were lots of volume. You had to administer 2 liters, 16 weeks of infusions, had to be in the ICU. So these complications really limited the potential utility of this approach, and it wasn't really widely adopted. And there was a lot of complications from the catheter system that he was using. And IL-2 is one example, but there's actually a lot of these additional molecular messengers that have potential. IL-2 is one that's already FDA approved for melanoma and renal cancer, but there are other promising ones as well that can be used, and they all have the same challenge where you want them to act locally. Systemic administration is difficult, and at the same time, systemic toxicities are a big problem. So we were inspired to think differently about this problem and leverage our biofactories to actually produce that drug interleukin right into that space. And by doing this, we were able to take that 2-liter infusion, turn it into about 5 to 10 cc's of these little beads. And they get administered right into the peritoneal cavity. And there's living cells that every day is producing the drug for a pre-programmed amount of time. So these last for 2 to 4 weeks, and then they stop making the drug. And we've been working on validating this in numerous animal models. One of the things that was really exciting to see was, the concept works. And in the peritoneal space, we're able to have this drug production, and then it stops. And in the blood, where you have all the toxic effects, you actually have very little amount of the drug. And you contrast this to traditional IV administration, where you get all these peaks and valleys. We're able to maintain that drug concentration for the duration that's needed. And we've done some work in mice and demonstrated that in these really difficult mouse models, we can eliminate these cancers. And we see really great results in terms of tumor treatment efficacy, protection against recurrence in these mouse models. But at the end of the day, they're mice, and we've also validated this concept in non-human primates and shown that we can achieve the right amount of the drug in the IP space. And in particular, if we contrast this to what Edwards was able to accomplish, we've tried different doses, which is essentially the number of capsules that get administered. And the dose that worked well for Edwards was about this 38,000 picogram per ml level. We're able to get there with our second dose, and the monkeys are actually no toxicity. We don't see any signs of toxicity, and we get the PD biomarkers in terms of the immune activation states that Edwards was seeing in his trial. So where are we today? As I mentioned, Avenge Bio is a spin-out company that has thankfully took a lot of hard work, but we were able to raise sufficient capital to support this clinical trial. And we've had our IND just cleared end of July, and we're working with different sites to get them up and running for the clinical trial. And what that trial looks like is 24 patients. We have 4 different dose levels. We've been working closely with Amir to design this and thinking about how do we get to the efficacious dose as quickly as possible. And then once we find the right dose, we would expand this to 24 more patients at that dose. And we're really excited to see how this turns out, of course, but really the first goal is to make sure it's safe and to allow us to understand the dosing responses and hopefully, patients see benefits. And, this has really made us rethink how we can use this in a number of other cancers, different molecules that we can produce. So we have a pipeline of different drugs. We're very excited about IL-2, but we also have other drugs in the work that leverages the same platform. And then I wanted to take, again, big shout out to Amanda, Amir, as well as some of the other team members, Samira and Maria and others who have helped with this. We also have we now have a team at Avenge Bio. There's about 50 scientists and business people and clinicians and everyone that's working closely on this. Mike Heffernan, our CEO, actually his wife had ovarian cancer as well, and that's one of the reasons why he was really excited about this opportunity and wanted to work on this with us. And then we have a really great team of advisors. Again, Amir, Katie Rezvani here locally in town, Raul, who's an interventional radiologist, because we hope to be able to administer these capsules as easy as possible using interventional radiology guided approaches, so you don't have to go into the OR. Yeah, but with that, I'm happy to, I guess we're doing questions at the end. So thank you so much.